Thank you guys all for coming. It's really nice to see such a great turnout. I think this is our first in-person seminar and almost at least a year and a half, if not 2 years. So thank you again for coming. I'm really pleased to have Dr. Abby Oliomi here with us today from Baylor College of Medicine. He got his PhD in Geographical Sciences from the University of Iowa. He did his postdoc at the University of Texas School of Public Health, which is where we first met. And then ended up, he's currently an assistant professor of epidemiology and GIS at Baylor College of Medicine. His research interest is pretty varied, but he is the GIS person at Baylor. So he gets pulled onto a lot of different projects, but most notably, he's done some microbiome and geospatial work. He's done some neat work on natural disaster response. I don't know if you guys have ever heard, but Houston floods a lot. He gets a lot of hurricanes. So he's done some great work and important work. And then today he's going to give just an overview of some of the different aspects of the work that he does. So let's welcome Dr. Oliomi.
Well, thank you. Thanks for the introductions, Dr. Nolan, and thanks to everyone. I should first ask if it's the folks on Zoom. I can't tell who's on Zoom. I guess I can tell who's not on Zoom, but folks on Zoom, I hope they can hear me, and I hope they can let you know. Let me also say thank you to the leadership team of the Big Data Health Center at U of SC. And I should also let you know, this is my first trip to Colombia. So I got here yesterday, and I think I'm gonna stay. We'll see. So, well, what am I going to do in the next about 40 minutes? And by the way, I should also apologize for the technical difficulties. Part of my fault because they asked for the slide. I didn't give it to them before. Anyhow, so what I'll try to do, with these masks on, is to it's not going to be things that are completely new, things that many people may have heard about before, but it's an attempt at trying to establish the utility, actually highlight and emphasize the utility of geographic information systems in biomedical research continuum. But I do that by, I'm going to just follow these 4 blocks. And what I'll do is I will start with the first block, which is to basically use environmental health, which in this sense is pretty broad, as the basis for which GIS can be optimally utilized in the biomedical research continuum. So as this, thing I just wrote that talks about the fact that basically any health condition you can think of, we all work in different areas. We all have different outcomes of interest, right? But if you think deeply about it, you can almost always link it to some type of environmental condition. And in 2003, right, they completed the Human Genome Project. And there's a particular person, Wilde is his name, C.P. Wilde, that wrote this, introduced a model that he called the exposome model. And the essence of the exposome model is to emphasize the need for a more complete environmental exposure assessment. Because now we have the genome sort of nailed down, right? Everything and anything we should know about DNA manipulation and epigenetics and all that. But his work was focused on the fact that that's not enough, right? So we need to be able to make the best use of what we've known, and when we realize it at that level, we need to be able to have a more complete understanding of people's exposure. And by that, it meant to be able to assess an individual's lifelong, at its best. The exposome model would be able to characterize an individual's lifelong exposure to all things environmental health, or environment, let's put it that way. And it meant, by environment, it meant non-genetic, so anything outside. So he basically created this graph to the right, this visual, as a representation. It's not exhaustive, of course, but it's basically saying, well, in the internal domain, you have these omics, and then you have specific external and the general external, which are going to be your socio-economic or socio-environmental factors. So his rationale, just to itemize, it's beyond these three, but these are three main ones. There's the fact that genetic variants in and of themselves provide limited understanding for disease risks, right? And the fact that there's a large role for environmental exposures and interactions among environmental and genetic factors. So you've heard about gene-environment interaction. So the essence is that if we can do this work and we can do it well, we'll have a more robust understanding of what's happening with the human condition in terms of health conditions. So I have this illustration, I'm going to show on the next slide, which is again, basic in many ways. And it's just to show how you can pictorialize the relationship between what is conception of the exposome is and epigenetic expressions. So, by the way, I'm no geneticist by any stretch of imagination. So I'm sure some of you know more about this than I do, right? Now, the thing is that we use it as an example, a situation where the histones, which are proteins that order the DNA and arrange them into structural units, can be affected by the reception that the histone tail gets, right? And these can turn off or turn on gene expression. The picture to the right shows two scenarios. The one in red is a situation where there are tightly bound histones arrangements, and the one to the right are loosely bound. Well, when highly bound histone arrangements, histones are present, you have more methylation going on. And that methylation, generally speaking, can indeed turn off gene expressions. But then we've also been able to at least appreciate the fact that these epigenetic processes may be influenced by nested exposures. And by nested exposures, we mean exposures in those big circles. Right? Concentric circles, interpersonal, community, environment. So we're seeing a situation where things happening at the genomic level or the gene level or the subcellular level, I think is the general term, are influenced by the things you can measure in the environment. And this raises two fundamental issues, right? The first one being the scales of influence, right? So now we're talking about DNA strands, things happening at that nano level, right? To things happening at the macro mega scale, right? The experiences you have in the neighborhood having effects on things happening at that very small level. I don't know if I still have, I used to have a little, box, but I don't think it's on this presentation. When I've given it before, I would ask people, what's our conception? Because when we talk about nano technology or nano level, it's a billionth of a meter, right? When you think about that, the illustration I had, which I don't think I have there, is a pinhead. So when you think about a pin, like literally needle, like a pin, a sewing pin, that pinhead, I would ask folks, how many nanometers you think that would be? But for the sake of being able to move forward, I would just say what it is, it's 1 million nanometer, right? So we're talking tiny, tiny level. So there is that issue of scales of influence when it comes to the exposome that we need to be able to address. And then the second one is temporality, right? The fact that, in fact, the exposome is saying, let's look at lifelong exposure from birth, right? So when you think about it, if you pick the environment as one of the higher level influences, because now we talked about scales of influence. You have the micro level to the macro, then you have to now think about temporality. So this person was born at a particular location. If they moved around, let's say 10 times in their lifetime, versus if they've lived in the same location for the entirety of their lifetime, their health outcome experience is going to be the same. Maybe yes, maybe no. The general sense is perhaps not. So because you have to think about scale of influence and temporality, I argue, and I think a lot of people would agree, that the exposome to be able to achieve its objectives would rely heavily on the power of GIS. So that's just the kind of like the first part, the fact that indeed for the exposome to succeed, it needs GIS. And this is just a little block to basically further expatiate on that point, that we're looking at factors operating at varying geospatial hierarchy and varying types. But that's not the only issue with that we have to deal with the exposome in general. It's an appealing model, but it's quite challenging, right? Because of this graphic to the right that shows you all the things you have to put into consideration in order to achieve the objectives of the exposure, right? The measuring, and I have just 3 listed at the bottom, which are supposed to be high-level challenges, right? The assessment of time-varying exposures can lead to misclassification, numerous measurements. Now we have the mobile devices. You're talking about questionnaire. You got biomarkers. These are things you need to integrate into a coherent form of analysis, analytical protocols. And then you got statistical challenges. Anyone that does statistics know that correlations is always the multicollinearity problem, the BIS and the tolerance and all that. So how do you deal with all those things? But at the center of this for this talk is the fact that even on this slide, you can see GIS as one of the components of the way to integrate the various levels of data that we're bringing into the exposure model analysis. So because I know people are coming from different backgrounds, normally I think in general when we give these talks and GIS, most people, I don't know how many people are familiar with GIS here, but I decided to put the slide here anyway. And if it's redundant, forgive me for that. But essentially, it's just a way to capture spatial data, process the data, display the data, and allow the data to help inform decisions. That's really what it's about, And but this little paragraph here to the right, under the graph, under the picture, is one of the things I like most about when explaining GIS to folks is that you can see patterns that you can't see in other programs, right? R actually has some GIS built into it. But in many cases, you still can't do as much as you would do in a native GIS protocol or environment. So, you see the graphic there that shows all the things you can do in terms of looking at the world and understanding how the world works. So that's GIS in a sense, just overall. So if you connect that with everything we've been saying, you would see how indeed this is a critical ingredient to being able to first assess exposures at different scales. Is it in your neighborhood? Is it in someone's neighborhood? Is it something happening at the city level, national, or and so on and so forth? And also to be able to look at time. The time element is definitely going to need geospatial analytics. And I have, I'll probably talk about some projects which you have a minute, where we're looking at time as a key component of the study. So So I kind of moved from the exposome, the environmental health importance to exposome, and how the exposome needs GIS because of the value of space, place, and time. So the next thing I'm going to do in this block is to just give you an example project where we did not design the project to do this, or these are essentially integrating data from various elements or exposure domains, but we're ending there. We didn't exclusively design it for that, but we're doing that now, and I'll show you some of the results. Now, this is, as Dr. Nolan mentioned earlier, Houston is a lover of floods. And this is Hurricane Harvey that hit Houston in 2017 that pretty much anyone probably in the world know about. And to what extent you all are familiar with the statistics, I'm not sure. But I like to just put this up just so people can appreciate the impact of the storm on the city. And you can just see all kinds of data points that are indeed worrying. But the one I actually would like to just highlight last, is the last bullet that talks about the amount of debris that we collected across the city, not just the city, actually, like the county, more or less. 200 million, this is one of those scenarios where numbers go really like, okay, what does that mean, right? So to contextualize that, I have it somewhere, but I think it's like maybe 100 or over 800 football, like big football, fields of debris, just debris. So anyhow, when that happened, everybody was, running around trying to figure out what's going on. The person in that New York Times article is actually my former head of my former research section. And we went and collected water samples, and there were calls coming into Baylor from different parts of the city, people asking for help with all the things that I lettered out to the right. So through that process, we ended up with a team at Baylor, at the UT School of Public Health, also in Houston, and then at Oregon State. And these are the tasks that every entity and the research centers have to manage. I was in charge of the GIS part of the project. So we put the team together, and we immediately wrote a grant to the NIHS that we wanted to be able to, I think I have here. The overall goals of the study was to create a cohort. So we wanted to create a cohort of people that were exposed to Harvey, and so we could follow them over time, assess immediate health outcomes, chemical exposures, and microbiome. If you think about the things we've been talking about since I started talking is looking at exposures across domains, right? So now we're talking about health outcomes, chemical exposures, and microbiome. And there's a slide that actually discusses that a little bit more. And the last thing is to be able to come up with best practices for future disasters. So those were the goals of the study. Specifically for the exposures that we assessed, and even outcomes as well. So we had a questionnaire that we designed to collect people's experience with Harvey. We used GIS actually across all buckets of the study, but then we collected questionnaire data, we collected biospecimens, which were to assess the microbiome, and then we got chemical exposure data. Folks had to wear, this is the work that the Oregon team does, is they actually created a wristband. It's like a regular, you think it's just a regular wristband, but it's a passive sampler. So when you wear it for a week, it collects up to 1,500 EOCs, volatile organic compounds, it's semi-EOCs. And so people wore those, they gave us samples, and then we got questionnaires. So those were the data points. And talking about the role of GIS, even from the beginning, when we set out to do the projects, we were intending to get people that were affected by Harvey. Even though Harvey was affected the entire area, not every, like I didn't flood, you know, I don't know where you sit there. Okay, I didn't. So everybody flooded, right? So, but we wanted to get people that actually flooded. So we used something called preliminary damage assessment that the FEMA puts out when an event like this happens. They will go and do surveys and assemble the number of properties, assets, or properties that have been affected, and they will provide the data at the census tract level. Now, this is another GIS peak, right? So census tract is what we generally consider to be a neighborhood, right? It's like a zip code. When you say zip code, but census tracts are smaller than zip codes. They're always smaller than zip code, I think, not always, but generally smaller than zip code. But they're designed specifically to mimic what you might consider to be a neighborhood. Zip codes are not. They're designed for efficient mail delivery. Yeah. So they do it in census tracts, and put it out. So we got that, and we used that data to create a map that showed us areas where people really flooded, right? So it was an iterative process, and we ended up with 4 neighborhoods, which are the ones you can see on the map. And just, you know, that's just to show you this little tessellations to the, this little hexagons show you the number of participants within a mile buffer. So that was all done in GIS and through GIS, and we went out and collected data. So fast forward, I mean, this was in 2017, right after the disaster. This was like September 2017. So we did it time one. I don't know if there's a graph that will show you that right after the disaster, and then we went back a year after to the same set of people and newer people too, to also collect more data. So we wanted to remember one of the goals is to see long-term effect. So this is one of the findings from the preliminary, the analysis we've done so far that shows the relationship between being flooded or being exposed to flooding exposures and allergic symptoms. And what this actually just shows is that a few, because at the bottom on the x-axis are the exposures, and on the y are the outcomes, right? So, and then of course you have 2 time points, 1 month after Harvey and then 12 months after Harvey. So just for illustration, when you look at molding home, folks 12 months after Harvey were more likely to complain or not complain, to report sinus irritation, even a year that were flooded, folks that were flooded, than those that were not. So that's one of the things we actually published this already. And then we also looked at stress. This is one of the fascinating things we found was that at 1 month right after Harvey, the level of stress that people felt, this is very generally speaking, but if you look at the data, it actually feels like people felt more stressed a year after Harvey than they felt right after Harvey. And there are all kinds of ideas about how or why that might be the case, right? Because maybe right after Harvey, were like, they just wanted to safeguard what they could, adrenaline fixing. But a year after, they've been able to go through processes dealing with the insurance company and all that, So, and we know stress has effects on health. I mean, that's an established link. The other thing I would highlight briefly on this graph is the fact that for people that were not, you see this little bar that comes, not little bar, actually, this bar that comes really down, are for those that did not have any exposure. So the people that reported that they had no exposure at all were significantly less likely to have reported stress levels, right? So, which is just confirming what you might expect, but to be able to see it in the data is actually fascinating. So we did that, that's global, that's everybody. I'm just gonna put this slide up and then I'll move on. We also looked at these same variables, variable stress, by socioeconomic deprivation. So part of what we do with the GIS protocols is we create deprivation levels for neighborhoods. So we can tell how deprived the neighborhood is. And we do that in GIS, and we use that to stratify the analysis such that you can look at areas with low ADI versus areas with high ADI. Guess what though? Areas with high ADI are it's counterintuitive, are the areas that are highly deprived. So they are, if you want to use like SCS speak, there will be low socioeconomic status area, if you use that terminology. So for low ADI at the high socioeconomic status area. So for whatever reason, it feels like people that are supposedly higher SCS maintain stress even 1 year after, which also has brought up a few ideas about, perhaps they've lost a lot of money and they feel stressed about it, but if you don't have that much money anyway, you may not be as stressed about it. So these are all kind of leading to all sorts of hypotheses that we're beginning to look at. But so I wanted to show you these analyses and some of the findings we've seen, but connecting all this back to the earlier point about the exposome, this is a little bit much, there's all these numbers. But what I want you to focus on is the fact that we got omics, we call them omics, microbiome, we actually refer to the chemicals as just the chemicals, and then in some cases we say omics. And then we have clinical outcomes that we got through questionnaires. So now we're beginning to look at different domains or several domains of data. Then the task is, part of what the exposome is saying is you got to integrate this data so you can see what's actually happening. It's not just one thing or two, it's maybe 3, 4, or 5. So I'm not gonna, because of time actually, because what I was saying here is to just think about, we have microbiome, this is gut. This is just gut microbiome. We're not even looking at nasal or saliva. So this is from a fecal sample, right? And the wristband data, which were 1,500 chemicals at time 1 and then at time 2. So you got the several exposure, that we talked about before, and then you got temporality. 1 month after, and then a year after harvest. And just so you know, we're actually doing a third wave now. We're collecting wave 3 data, which is 2021 versus 2017. And we've already gotten of the people we had at time when we had 173, 266, because we added more people at time 2. Now we have 106 that are in the study still. So you can imagine that we will have another extend this graph, and then you have the same set of variables. The only thing we don't have now is the wristband, actually, but we have everything else. So just to wrap it out, to show you what I'm not going to spend time on that one, but yeah, it's this one. So this is one way of looking at the data, right? It feels like someone got crying. But I just put boxes around 2 of the graphs. Essentially, what the first A is saying is that there is significant difference, or there are significant differences across the study area. So talking GIS, now we have, you remember we have 4 neighborhoods, right? So East Houston, we should have put the graph there, sorry, but you can't say, but the first one is Addicts, Baytown, Bel Air, Maryland, East Houston. And then we have some people that just came from all over, right? They weren't in any of those 4 neighborhoods, even though those are the areas we targeted. So we ended up with, not too many, but they were just from all over the county. So we put them in their own category. So you can think of them as the people that are just randomly existed, right? So if you want to, you can say, well, you can use them as a standard that you're comparing the neighborhoods. Anyhow, so but the A essentially is showing that in East Houston, which is this one right there, that the concentration of the chemicals that we detected, we didn't detect all 1,500, by the way, are generally higher in that neighborhood versus the other neighborhoods. That's one way of interpreting that box. Although Baytown too has some, but they're a little different. The one to the upper band of the graph. And then this is the pathway, the microbial pathway, the microbial pathway for across the study area again. And you can also see each system is showing some interesting coloring here, where the upper band has more abundant microbial pathways, and then at the lower band is showing less abundant microbial pathways. These are we haven't gotten into which ones are the chemicals or which ones are the pathways. We're just saying this is what we see in these areas, right? Now we may need to go down and say, okay, what's so high in these two states and why? So we're beginning to ask that question now. And one of the ways we're using GIS in that realm is to look at proximity to chemical plants, Superfund sites, and other points of interest, including actually dry cleaning locations, right? So because we've seen this, we can identify the VOCs that are higher. Then we can now go say, well, what's happening in terms of locations using GIS as a way to do that, which you couldn't do without GIS. So that was another graph. I took that one out. That one. Even I don't understand that one. No, I don't do this. By the way, we have a lab. It's a bioinformatics lab. They do this work, and I just talked about that. So that's just. I wanted to give you that as a sample project, in an attempt to show how at least we're trying to. We're not even living up to the exposome ideals yet, because we're looking at 2 time points, right? We're talking about lifetime, and so on. But at the same time, having sort of explained all that, in being fair to the title of the talk, right? The fact that that's just one way of exposome and GIS, but then how does that connect to the biological research continuum? And they do connect, and that's what I would attempt to do here. Now, hopefully I'll succeed, we'll see. But, so what's the biological research enterprise? It's actually what I like the word enterprise a lot, because that's from the NIH, by the way, is the fact that you got, so when people say biomedical, the first thing that comes to many people's minds is labs, white coats, right? Mice and mouse, right? Mm-hmm. But indeed, the thing I underline is really what I like to focus on, is that whatever it is, whether it's a mouse or a questionnaire or our package, whatever it is, it's all toward a goal. And that goal is to improve human health and longevity. If it's that, and you're doing it scientifically sound way, then it's biomedical research. So it's many fields, many scientific fields and all, right? So that's the enterprise. Well, the enterprise, in a sense, can now be described, articulated, understood, use any adjective, through the translational science spectrum, right? Where you start with basic science research and you end with translation to community, which is right, you have an impact on the community level. And even when we talk about that sort of spectrum, we tend to think of it as a linear spectrum. Like you go to start everything from the lab. That's why you do a discovery, and then you experiment, you see if it works, and then when it works, then you're good. You can go put it in. No, it's actually multi-directional, not unidirectional, and it's not linear at all. You can actually have discovery at a community level research, right? So that's the other thing. So think about biomedical research continuum, and then think about it in terms of translational science spectrum. Then the next question now is, how does GIS fit into that spectrum? right? But we can, I mean, I think many of us can easily connect GIS with the upper, you know, part of the spectrum, right? The T3, T4, you know, that's where you really just working in neighborhoods, right? So, well, we can, the best way to articulate what's going on in a neighborhood is to be able to characterize that neighborhood. To characterize a neighborhood, you need to be able to do spatial analysis. Well, you can ask me, you know, so you can ask me, you can come to me now and give me a questionnaire and say, oh, think about your neighborhood. Do you think there's high crime? And I'll say yes, right? Or no. I mean, I don't know. but I can also not ask anyone and just go to the police record and pull all the files that they're doing right now, and locate where the, you know, the stuff happens and create a, you know, assessment of the neighborhood. So I guess I'm saying you can ask people, you can, you know, GIS is not the only way to do it, but GIS is one way to do it very well. So that's easy. We did something, so I show an example of how GIS would apply adaptive T4 level, and it's a recent issue. I mean, it's an ongoing problem. So I'm wearing a mask, right? So it's COVID, right? So the story of this project is that when COVID really began to really pick up in the US, we were scrambling, right? I mean, people went to show exactly what to do with what, you know, even with data set. But bottom line is, in this case, in May of last year, the county decided to put up you guys probably saw a lot of those dashboards. The most popular one is the Johns Hopkins one. Everybody used it. By the way, you know, so I'm going to tell you. So we went to their back end and pulled their data on time series. This is Johns Hopkins data. Maybe they're listening. and maybe we don't have the right data set. So they say every day in the US, they tell the number of people that got COVID at the county level. It's, you know, every county. We have about over 3,000 counties in the US. So they give you the number. County, day. So it's a large data set. It's like daily for almost 2 years, 3,000 rows, you know, calculate that. It's a matrix. And we thought, okay, well, we want weekly estimates. That's what we wanted to use for analysis. We want to do a lagged analysis looking at cases and death across the US. Guess what? We tallied the numbers and I started saying like minus death. So what happened was, in some cases, the number for the day after was less than the number for the day before. You know, and you know, I say that not just to say that Johns Hopkins is not great. I mean, I'm sure they did a great job, but it's also to highlight the fact that when we're talking about big data, which is what the center is about, and GIS, you have to be extremely mindful and careful about how you tally data, get data, process data. I mean, if we were smaller numbers, we may not have seen, right? If it just was less by less, you know, and then when we sum the weeks, it would disappear, right? So that's just one quick note. But anyhow, to the slide, the county gave us the data at the zip code level. They put out these things at the zip code level. Zip codes, again, are not useful, not very useful. Because they're for mail delivery. So when you're talking neighborhood, they're not the best things to use. Sometimes that's all you got, and then you have to use it. But if you can get anything that's less than zip code, please get it. So bottom line is, we use GIS to go from the zip codes to the census tracts, right? And that could be so tall. But the bottom line is, we went from this map to the left, which is zip code, to this map to the right, which is census, but it's a model, right? So this is actually observed data, but this is model. It's a prediction. I mean, it's not prediction, it's model. We use the prediction surface. So that way we can say, well, in this neighborhood, this is what COVID looked like, right? And then the next thing we did was to say, okay, well, within that neighborhood, what are the things that were associated with higher COVID cases? This was in the beginning, where everybody was trying to figure out what's going on. And we saw those things. We started with 29 indicators. We ended with 7 or so. One of them got kicked out because you don't know. So this is what we ended with. Those are the IRRs. 2 had less likely to have high COVID. And then the next thing we did in GIS is to now, from an intervention perspective, say, okay, these are the things associated with high cases of COVID. Are there things we can modify or we can intervene on, right? Not we, but the council. So we did this to the right, and that's your, so that's the geographically weighted regression that estimates the IRR, the risk, for each neighborhood separately. So now you can say, oh, if I look at, what can you change? Okay, households with no vehicles, right? If someone came to Harry's and say, we got, 10,000, electric Teslas that would like to get people to be able to go, around and get tested, so we know. If you want to do that, you're better off here with that intervention than you would be here, right? So we can use GIS to actually make that kind of decision. You have to make assumptions about how you build the model anyway, but this is one way of showing that. So back to the translational research, we're using GIS at the T3, T4 level, because this is all T3, T4. This is community level and all that, right? And then so I jumped out, I was also talking to a green in between, but I just jumped all the way back to the basic, because that's a place where you least expect GIS to be useful, is in the lab, right? Like, okay, what are we going to do with that? So, but you still do something, and in this case, I just wrote up what we're working on right now. It's very preliminary, so there are no data. I can't talk too much about it, but the larger point is that they found a lab, they had novel findings that showed alterations in DNA methylation that was distinguished or differentiated between a group and another group. And the question is, remember that graph that goes from the nano to the macro? Like, are there expressions in the neighborhood or the larger environment of those individuals that we can actually use to understand the mechanisms that dictates the differentiation? So essentially, are there things happening at the neighborhood level and other levels that affect what we're seeing in the lab in terms of epigenetics, right? So this is something that will happen in 2 states and all that. But so that's why we can use GIS to help us with that. So I think I want to be able to have that talk, to have questions. So this is just a bunch of points about what I try to cover. But I'm not going to go over them now. But I think that's pretty much it. So I will leave it. I don't know if I'll be able to come back to see the last thing, but let me just say it has recognitions for people that have been on the team, and then I've sent several other HBC faculty, and even people faculty across different institutions in the US that we work with. But so that's it. So thanks for allowing me to go on and on for less than 50 minutes, but I want to be able to have to ask questions if they can. And I see a bunch of those.
Oh, those were from earlier.
Okay.
But people attending online can type their questions in the chat. So you can keep that pulled up if you want to be able to see what they say, or I can read them off.
Okay. Yeah. Okay. So thank you.
I'll start. For those students in here, can you talk a little bit about spatial analytics that they could possibly use? Like, so you mentioned geographic weighted regression. What is that?
So in the example I showed, which I think probably in any normal, well, yes, a typical analysis, right? Okay. Okay, before we show that. So this is the table at the bottom is what we all do, right? So we take indicators or explanatory variables, X, and then we have the Y, the outcome, and we run whatever analysis you're running, whatever regression, Poisson, and you have your effect estimates, right? It could be odds ratios, it could be, in this case, IRR, relative risk. But what the analytical protocol is doing is saying, essentially saying on the average, right? Mm-hmm. So it's saying, in this case, we say a % Black or African American population in a neighborhood, right? When that increases by 10 units, because it's a 10 unit, okay, 10 unit increase in the % African American Black population increases the risk by 1.02. Right? Now, the GWR goes past that. It says, this is it for African Americans and Black. It says in these areas, in Harris County, right, I know that globally, % African American is associated with increased risk, but I'm going to tell you what parts of the county that is more so the case. So whatever the finding is at the global estimate, it's going to tell you where it's emphasized, de-emphasized, or going the opposite direction. So in some cases, it actually there are areas within the county where that indicator would actually be protected rather than be a risk score. So that's what the GWR does, allows us to do. Yeah, so that's one way you can use GIS in terms of the question, and especially for GWR. But you know what we do with GIS at IB is actually not geostatistics. You know, it's geodata processing. So allowing, giving us the opportunity to process and create variables using GIS, right? So translating data from spatial sense to text, to numbers that you can put in your model. So that you can do proximity analysis, you can do overlay analysis, but proximity, like I said, we're looking at distance to superfund sites. That's not geostatistics. It's just, you know, we're just going to calculate the distance. But we're using a program to do it. Overlay would say, okay, like we did with the same situation, we say, there's a project now where we're looking at the density of dialysis centers across the US, because we want to use that to understand the utility of telemedicine during COVID. So we got all the dialysis centers in the US. Now to do that, we're talking about density. So we say, well, we're going to put in a census tract, count the number of dialysis centers, and then divide it by some indicator, right? Like normalize it by population, right? So, you know, you do overlay analysis, you can do all sorts of things in GIS, if that helps. Oh, yeah.
Fantastic presentation. I've learned a lot about, especially talk about regression here. I'm just curious, the spatial units is really important right here to look at the differences across different areas in terms of some variables. And how can you identify the smallest unit? I'm just curious to say neighborhood, the probable neighborhoods are really good indicators to get some kind of outcome, for example, COVID-19. But if you look at an artist's outcomes, how can you identify and can you provide some type of guidelines to understanding how to do the decisions of that?
Okay.
Thank you.
No, thanks for a great question. So in this case, so that in terms of the smallest neighborhood, there is a standardized, sort of using standardized geography, which is what this is. We say, okay, we know, because we know that it's informed knowledge that zip codes are not good for, therefore, mail delivery. There are geographies that the census provides, right? And that's census tracts, block groups, blocks. Blocks is the smallest unit you can get at the census tract. But because it's so small, the census doesn't release all the indicators for that unit for confidentiality reasons, right? So they don't provide a bunch of the variables that we use at the block level. But then at census tract level, they will. Because, you know, average population for a census tract is between 4,000 and 6,000, which is small, right? So we go that route, we use standard geographies. But there are instances where for specific outcomes, standard geography would not cut it, right? Which is, I guess, what you're saying. Then you now start to think about what's the best unit that I should use to quantify my exposure. In that case, then you have to go to geostatistics. I mean, that's the only way I know is you now have to be able to translate, because you got to get the data. If you're not going to go ask a question or measure it yourself, if you're using existing data set, then you have to now use small area estimates analysis. And that's combining what you have right now with geostatistical models to tie it into the specific geographies that you want to create, right? So that's the only, so you either use the standard geographies like we did here, or which is easy because the data are already assembled at that geography, or you use small area estimates, which is statistics actually, to create new units of analysis. And by the way, the CDC did that with they did it with some other nonprofit. It's called 500 cities. It's called 500 cities. So 500 cities is a project that, and I think they've been updated to estimate specific health outcomes at the census tract level. So like cancer, some, not all cancer, but some cancer, some behavioral outcomes, and they use small area estimates to do that as well. So, so those are the two ways I could think of that.
No questions from our rather shy online audience. So.
Thank you for the great talk. So I'm in a totally different field, but I really like your thought on, lifespan perspective. Yeah. Like when people are moving around their life, they're actually exposed to different environmental factors. Yeah. But I'm also thinking about the other way around, like even within the same lifespan, like if someone is just staying in Colombia for his or her whole life, yeah, their exposure to this environment, those factors could also change. Yeah. So the other way around, like what's your thought on how long it would take for an environmental factor to predict or impact someone's health outcome?
Cool. Yeah. No, that's a fantastic question. And I was having a conversation about these with someone yesterday. So there's a project we're looking at HIV incidence and ADI over 7 years for VA population. And exactly the question was like, okay, so we're looking at when they were diagnosed, what's the ADI? I talked about ADI, this is the definition, at their neighborhood of residence when they were diagnosed. And then the question was like, okay, so how does this thing change? Gentrification is just a way of appreciating it, right? So if a neighborhood goes from nothing to like the most expensive part of the city within 3, 4, 5 years. So the best I can, there's no, I don't think there's I don't have like an answer. I think it's a very interesting, not just interesting, it's an essential question we've got to start dealing with how do these things, how do neighborhoods change, and how do those changes, whether people move or not, right, affect them where they're at. So you're totally right on that. And it's not just people moving around, it's also being able to continuously update assessment of the same area again and again. Thanks.
Yeah.
And yeah.
So if there's no more questions, I want to thank the virtual audience, and I'll end it here, and then I'll give you guys.
Thank you.
Thanks, everyone.
Thank you.